tiny titans, unveiling the microscopic world. Bacteria, invisible to the naked eye, are titans of their own realm. These single-celled organisms, numbering in the trillions, inhabit every corner of our planet. Understanding bacteria, their size, and their behavior is crucial to fields like medicine, environmental science, and even food production. But how do we study something so incredibly small? The answer lies in the magic of microscopy. Microscopy opens a window into this unseen world, allowing us to observe and measure these tiny life forms. By understanding their dimensions, we gain insights into their growth patterns, responses to environmental changes, and even their potential to cause disease. In essence, measuring bacteria is not just about size, it's about unraveling the secrets of a hidden universe. Windows to the invisible, the power of microscopy. Microscopes are powerful tools that extend our vision far beyond the capabilities of the human eye. They use lenses and light to magnify objects that are too small to see otherwise. For studying bacteria, light microscopes are commonly used, illuminating the tiny cells and making them visible to us. There are various types of light microscopes, each with its strengths. Some use bright field illumination, providing a clear view of the bacterial cells against a bright background. Others, like phase contrast microscopes, enhance the contrast between the cells and their surroundings, making them easier to observe. Regardless of the type, microscopy is essential for exploring the hidden world of bacteria. Preparing for the microscopic stage, a step-by-step guide. Before we can measure bacteria, we need to prepare them for viewing under a microscope. This preparation involves a series of steps to ensure that the cells are clearly visible and properly preserved. The first step is to obtain a sample of bacteria, which could be from soil, water, or even a laboratory culture. Once a sample is collected, it's often spread thinly onto a glass slide, a process known as smearing. The smear is then air-dried and heat-fixed, which kills the bacteria and adheres them to the slide. Next, staining techniques are employed, using dyes to color the bacteria and make them more visible under the microscope. With the sample prepared, we're ready to delve into the microscopic world. Measuring the unseen, quantifying bacterial dimensions. Now comes the moment of truth, measuring the bacterial cells. Using a calibrated microscope, we can determine their size. The microscope eyepiece contains a ruler-like scale called an ocular micrometer. By comparing the size of the bacteria to this scale, we can estimate their dimensions. However, to obtain accurate measurements, we need to calibrate the ocular micrometer using a stage micrometer. This device, with precisely etched divisions, is placed on the microscope stage and aligned with the ocular micrometer. By comparing the two scales, we can determine the actual size represented by each division on the ocular micrometer. With this calibration, we're equipped to measure the bacteria with precision, revealing their true dimensions and providing valuable insights into their world.